All right. Good morning, ladies and gentlemen. My name is Jirat Phoonphon Jeerangkul, and today I'm going to present the e-poster on the topic of decoding the impact of RGL genetic variants on protein structures and binding site, bioinformatic approach to uncover potential hypertension association. So let's start with the background. The RGL3 has been identified in many studies, especially the Genome-West Association study, that it's associated with hypertension. And interestingly, its loss of function from the normal function result in a lower incidence of hypertension. However, no study mentioned how or why. So we would like to use the bioinformatic approach to assess the change of the protein structures resulting from this mutation. Now, in terms of method, we acquired the clinical variants data, which means the sequence from the ClinVar database of NCBI. And after we retrieve the variants, it categorized the variant to two parts, the likely benign part and the uncertain significance part. We're going to focus more on that. And after that, we do the multiple sequence alignment, which means we're comparing the normal and the mutated variants by using the bioedit tools. And after we know where exactly it is from the sequence alignment, we construct 3D models from it, comparing normal and wild type by using the AlphaFold tool, the AI that's got its Nobel Prize this year. And we modified a little bit by using BioDiscovery Studio visualizer tools. And after that, we assess the quality of the 3D models by using the project. And lastly, after we know how does it look, we assess the physical chemical properties of the amino acid in each mutation. Now, the result, we got 81 variances, and 5 of them are likely benign, 76 of them are uncertain significance. And according to the sequence alignment, interestingly, all of them are missense point mutation, which means that only one nucleotide change, and it changed the amino acid from one to another. And one can see that this is the sequence alignment. The highlighted one is the amino acid that changed. It's scattered throughout the variances. And after we know where it is, we construct the 3D models. This is the normal on the left and the mutated on the right. The colors represent the binding site or the domains of the protein. The first one, the yellow domain, is the exchange N-terminal domain, which interact as or with the RAS protein and act as a signal transducer. The red one, the middle one, is the most important one here because it's a catalytic regions which act as changing from GDP to GDP in RAS GDP, because the purpose of this protein is to change from RAS GDP to RAS GTP. And the last one is RAS association, which bind to the already activated protein and do its further signal transduction. And this is the plot called Ramachandran plot. It assesses the quality of 3D model. It tells where exactly amino acid is supposed to be. Red is it really supposed to be there. Yellow means it's supposed to be there, and white area is not supposed to be there. And one can see that not many amino acid in the white part, and the numbers of amino acid in this around region is less than 2%. It's only 0.5, which means it's actually a great model. And this is the summary of all the mutation. It's quite extensive, I know, but I would like to emphasize on the most important part is that we categorize them into three groups: most important, moderate important, and least important, by using the property change of the amino acid. For example, the first one, tyrosine to cysteine, it changed from polar to non-polar, and it changed drastically. So that's why we say that it's changed mostly. And this is the one can so-called the criteria of the property change. For example, the charge, the polarity, aromaticity, size, flexibility, and hydrophobicity. And if there's a big change in this, we could say that there might affect the bindings or interaction of the protein and might affect the functions of the signal transduction pathway. So in summary, this protein act as one of the protein that help in signal transduction, and it involved with many pathways, for example, MAP kinase or maybe PI3Ks. And if there's some kind of mutation in this protein and affect its function, it could lead to many things. So this particular research opened a possibility of many more research that assess the functions and the quality of a protein that result from this kind of little but could be important mutation. Thank you.
That was a nice topics and presentations. Are there any questions or comments from our judge?
Oh, so you have a select to use a many variant, right, for using in your work, and because the hypertension is quite uh, broad in many different regions. So, what is your criteria to select the variant for your work, and is this variant is specific for national or international?
Okay, thank you for your question. So first of all, we select the genes from the study since it beginning from us reading the papers and these genes what mentioned in many GWAS paper, and after we literature review more of it, it's not really a common gene. It's actually quite new. So we go to the database and we actually gathered all of the variants from the database. And unfortunately, it does not say that where exactly from what kind of population that it from, but I would say it's quite international because there are several studies that published in the database. But like I say, it does not say about the quality wise of the variants.
Could you specify roughly how many European people or how many Caucasian or Mongolian in this
Yeah.
study in
Well,
percent?
well, they did not mention that, and I'm sorry that I cannot answer that question. So it might be a possible but and crucial study in terms of population genetic first, and if we have that, we could maybe do this again, and it might show something more really interesting. Yeah.
We have the questions from Dr. Bandit from
Okay.
online.
Yes. For the RGL3 genes, specifically, is it specifically for the hypertension, or could it be implicated in all other diseases? And if you want to design the drugs or anything targeting these genes, would it be affects all other systems? What do you think?
Oh, okay. So it's actually related with hypertension and also hypertension in the pregnancies. For example, the preeclampsia and eclampsia. And I would like to say that the further implication, like at the very end of this study, it would not be really like a therapeutic target because one does not use gene therapy for hypertension, right? But it's more like a screening tool as a preventive medicine. Since we know that one have this gene, we can like prevent the hypertension or maybe preeclampsia in the patient. Yeah.